In the second half of the 19th century, for the scientists, it became quite evident that there is something which separates the inside and the outside of the cell. And in 1877, a scientist named Pfeffer was the first to study the so-called osmotic pressure within cells and formulated the idea that the cytosol of the cell is surrounded by a thin layer which he called plasma membrane. So Pfeffer in 1877 was the first to introduce the term plasma membrane for this thin layer he proposed present on the surface of the cell and surrounding the cell and separating the inside from outside. During the same period of time, another physician, Charles Overton, this scientist and physician as a anesthesiologist in a hospital, and indeed that's the beginning of the introduction of anesthesia during surgeries with the discovery of ether and chloroform. And because ether and chloroform are lipid-soluble substances, Overton proposed that by observing how these lipid-soluble substances penetrate into the cells of the brain and achieve this anesthetic effect or inhibit the transmission of nerve impulses between neurons, he proposed that lipid-soluble substances penetrate readily into the cells, whereas water-soluble substances do not. And because of this, he first introduced his theory that the plasma membrane represents a lipid-impregnated barrier layer. So Overton is the first to assume that probably plasma membrane constitutes of lipids because it readily dissolves lipids and allows for lipids to penetrate into the cell. And in opposite, water-soluble substances have huge difficulties penetrating the plasma membrane. But this was only an idea. There were no evidences, scientific evidences, for the presence of lipids in the plasma membrane. At that time, or close to this period, another scientist, not physician, he was physicist, working separately on his projects of studying lipids, made something very special. This scientist is named Erwin Langmuir, and in 1917 he constructed a very special device on which you can make a monolayer of phospholipids. As a physicist and chemist, he was studying the behavior of phospholipids, and he made this special device on which you can make a monolayer of lipids at the interface between air and water. And Erwin Langmuir was not studying the plasma membrane and its features, but this device he constructed was used by other scientists, namely Gorter and Grendel, to make their model of plasma membrane. The model of Gorter and Grendel, they postulated this model in 1925 by performing a very beautiful and clever experiment. They used this device of Langmuir What was their experiment? They take blood from different animals, from cat, from sheep, from goat, from rabbit, even from human. And you know, in blood there are many cells. Most of them are red blood cells, erythrocytes. They are the dominant cell type. And what they did was to extract chemically the lipids from the cells, from the plasma membranes of these cells. And then, using the Langmuir's device, they spread these lipids in a monolayer on the interface between water and air. And on the device of Langmuir, you are able to measure the surface area of this monolayer. Well, they have collected a dedicated number of cells, and from this dedicated number of cells, they have extracted lipids. They can measure the surface area of the cells and the plasma membranes. And if you extract lipids and spread them in a monolayer, you expect to receive the joint surface area of the cells. But without the acceptable error from their experiment, when they spread the lipids in a monolayer, what they observed is that the monolayer is almost larger than the joint surface area of the cells from which they extracted the lipids. And because of this, and because this experiment replicated in different animal species, they postulated that plasma membrane consists of a phospholipid bilayer. And you see here the simplified diagram which describes the model of Gorter and Grendel. These molecules that you see here are a representation of phospholipids. That's how we draw phospholipids in chemistry. This is the polar head, which usually has some charge, in most cases negative, and then this phospholipid is attached to two fatty acids, which are uncharged molecules, and this is the polar phospholipid with hydrophilic heads facing outside and inside, and hydrophobic tails facing towards each other. So this is the model of Gorter and Grendel. But this model has had many difficulties explaining most of the functions of the plasma membrane, which I described at the beginning. Well, lipid bilayer represents a quite efficient physical barrier, and it has some selective permeability, but yet proteins and charged ions can penetrate some plasma membranes even faster than lipid-soluble molecules. If this is only the valid model, so this lipid bilayer will be unpermeable for charged molecules or ions, which also have positive or negative charge, and also large proteins and other polar molecules will have huge difficulties penetrating this layer. But in most of the cells, ions and large charged molecules penetrate the plasma membranes quite readily. So there must be something else present in the plasma membrane together with phospholipids. And here comes the next model, which was postulated separately by Daniel and Davson. We know the model of Danieli-Davson, postulated in 1935. This is the model of the sandwich. They said that lipid bilayer is also having proteins located there, and these proteins are located on both sides of the lipid bilayer as a sandwich. In the middle you have lipids, and On both sides, you have a layer of proteins, and this is the sandwich model. But this model was quite simplified, and it cannot explain why plasma membrane serves different functions in different areas of the cell. So plasma membrane is not equal around the cell. It has functional domains around the cell. And to explain this function of the plasma membrane, in 1972 came the model of Singer and Nicholson. But before that, with the invention and introduction of electron microscopy to the cell biology, a very prominent scientist, Robertson, studied in detail different membranes, not only the plasma membrane, but also the membranes of organelles, membrane-bound organelles. And when observing the plasma membrane under electron microscope on sections stained with osmium tetroxide, he saw something very interesting. Plasma membrane was made by a dense, electron-dense layers on both surfaces, intracellular and outer surface, and between the two dense layers, electron-dense layers, there was an electron translucent layer. And you can see here, these are two cells in contact with each other. This is the cytoplasm of one cell, and here is the cytoplasm of another cell. This is the plasma membrane of one of the cells, and here the plasma membrane of the other cell. And you see the black electron dense layers on the surface of inner and outer surface of the plasma membrane, and the translucent layer between them. And this representation of the plasma membrane under electron microscope appeared to confirm the model of sandwich, the model of Danieli and Dawson. We have the protein layers, which are binding osmium tetroxide and appear black, and between them lipids which are not attracting osmium, and the lipid layer appears almost translucent on electron micrographs. And what is more, by studying the plasma membrane and membranes of membrane-bound organelles in the cell, Robertson found that this structure is replicated everywhere. So he introduced the unit membrane theory. What postulates this? All membranes in the cell, plasma and organelle membranes, have the same structure. A bilayer of phospholipids with monolayers of proteins on both sides of it. And until that time, everything was thought to be clear about the structure of the plasma membrane. But as I said, this uniform distribution of proteins could not explain differences in the function of the plasma membrane in different domains. So everywhere the plasma membrane has something different related to its function, but function comes from structure. So if the function differs, structure also must be different. That's why a few decades later, in 1972, Singer and Nicholson came with their model of plasma membrane, which is the valid model today, and this is the fluid mosaic model of Singer and Nicholson. As I said, that's the dominant model today, and it describes the plasma membrane as a lipid bilayer of phospholipids and cholesterol, in which proteins are floating like icebergs in the ocean. And we have some proteins which penetrate the whole lipid bilayer, and these are called transmembrane proteins. And we have proteins which are associated only with the outer or inner lipid bilayer, and these are called peripheral proteins. And these proteins can float, in the lipid bilayer, literally like icebergs are um, floating in the waters of the ocean and moving and changing their position in the plasma membrane. So that's the current model describing the structure of the plasma membrane. And now we'll see in detail what this model postulates and which are the building blocks of the plasma membrane. Uh, Plasma membrane, as we know,